I will now discuss on a very interesting topic and fascinating topic, that is MEMS for biomedical applications. And you know, if something is used for human body for biological application, so you have to take 100% precaution before using it in any of the animals or any of the biological system, because you cannot make harm to the system for your devices which you are intend to use for certain application. They may not deteriorate the functioning of the existing living beings. At the same time, you should not do something which will create some problem in the environment also. So that is why, like other sensors which are being used in case of avionics, in case of automobiles, in case of say space, or in case of industrial control or entertainment or IT, whatever it is. So those sensors are, I agree that those sensors are high precision and good quality, but those are not dealing with directly the biological system or any of the living being organism. But if you go for using a particular sensors and actuators for living beings, so he has to qualify other aspects also. Those aspects are mainly it has to be biocompatible. But what is that? That is, if you are going to reduce certain diseases or remove certain diseases in a living body or human being, so the methods you are going to adopt, they cannot call for other problems in the human body. So that is the point you have to take care. So that is why in case of biological system, the MEMS which are coming up for biological application, real life practical testing is becoming more and more difficult until unless you get permission from different government bodies or authorities who control the biological medicines or any kind of the biological interaction. So their clearance is required before you go for testing of those devices. So we will just see now in this lecture how the MEMS device is going to play a major role in different biological functioning and how it can be used for diagnostic purpose in a fascinating way, in a very challenging way. Now, if you look into the BioMEMS, biological MEMS application is sometimes called is a BioMEMS. So in BioMEMS, what are the different things coming into the picture? Here you can see, first is the micro nano fabrication, and in the middle is that engineering technology and life science. Circle is there in the life science and engineering technology we are adopting, and now lot of permutation combinations are going on, lot of evolutions are taking place. And what is the input to this evolution, to this research? One is materials and interface, another is chemistry and microbiology, and characterization technology. So these are the various inputs. Micro and nano fabrication is the basic thing, because of which you are getting the MEMS sensors, and then evolution is possible, that means improvement. Evolution means in positive direction. For that you need thoroughly the knowledge or understanding of materials which you are going to use for biological application, and they are interfacing with the biological system. Chemistry you have to know, particularly microbiology, biochemistry, you have a good knowledge on that, and characterization technology. This characterization technology in many cases should be non-evasive. So those kinds of things you have to keep in mind. So all these things makes the BioMEMS area more challenging. Now biotechnology encompasses what one is micro and nano fabrication, as I showed you, MEMS and NEMS, micro electromechanical systems, and NEMS is nano electromechanical systems, biological and chemical systems ranging from cellular to molecular level. You have to go to the cell level, what chemistry is going on. Molecular level you have to see something. If you go for DNA research or gene research, then you have to understand the microbiology fully. Techniques and materials to interface the biological and synthetic world. So, interface is another point, because ultimately diagnostic and controlling part will be electronic, but which you are going to implant the devices into the human or any living body. So, mostly they are synthetic or polymeric in nature. Many of the metallic structure you cannot use inside the body, which will deteriorate, which will react with lot of systems. And characterization of such hybrid bioelectronic system. Bioelectronic system characterization, how do you get output of that and how would you modify those things? For that you need the characterization of the complete system. Now, what is the definition of BioMEMS? Lot of scientists in that area, they put forward a lot of definitions. I have quoted some of the definitions here. One is from Basir from Purdue University. According to him, the devices or systems constructed using techniques inspired from micro nanoscale fabrication that are used for processing, delivery, manipulation, analysis or construction of biological and chemical entities. These are BioMEMS. On the other hand, some other scientists in 2000, they defined biosensors as their analytical devices that combine a biologically sensitive element with a physical or chemical transducer to selectively and quantitatively detect the presence of specific compounds in a given external environment. This is another definition. So, ultimately what biochips can do? Biochips can detect cells, microorganisms, viruses, proteins, DNA and small molecules of biomedical importance and interest. So, your system should be such that the microsystem or microsensor, they can detect all those kinds of things. What are the applicant ranges of BioMEMS? In case of diagnostics such as DNA and protein microarrays, microfluidics, tissue engineering, drug delivery systems, implantable BioMEMS, these are different application ranges of biosensors. The working environment of the BioMEMS is shown in this particular picture, where you can see some extra precautions has been taken to protect the workers or researchers, because you are dealing with some of the chemicals, some of the viruses which may be dangerous to mankind. So, if you are dealing with those viruses or the chemicals or proteins, whatever it is which may be harmful to human body, you have to take that precautions. Those who are working on the gene therapy or the genomics and proteinomics, gene transfection, so they have to take care a lot before handling those kind of the elements for characterization and testing. Now, before going to little bit in detail of the sensors, here are some of the BioMEMS devices which are marketed. Those are shown in this slide. First one is from University of Michigan, second one is from University of Stanford. This is a neuroprobes for neurological application, and third one is Max Planck Institute. These are some of the cells for testing certain diagnostic purpose. Here from Cumetrix, some player in the BioMEMS area, you can see they have devised a microneedle, and this microneedle, and in the other side is the hair, those dimension, if the tip of the microneedle is smaller, much smaller than the tip than that of the hair dimension. So, that microneedle has been fabricated using silicon micromachining technology. Some silicon biochips have been reported from Purdue University, that is also shown here, and for IBM Zurich Research Lab you can see some of the cantilever structures which are used for biomechanics also. So, these are some of the research results from various laboratories around the globe who are working since last 5-10 years on various areas of the biosensors. Some other finished product from companies are shown here. One is the cartridge, biomedical cartridge from Nanogen. Some is immunochip. This one is immunochip from Aclara. Here is PDMS glass microfluidic biochip, and this is lab on chip from Calipar. So, these are some finished products from the companies. Now, what are the different aspects of BioMEMS? One is materials, obviously, and in biological application, silicon, although it is used, but it is not a good choice of material which can be implanted into the human body. Some other microelectronic materials people are looking for, those are glass and quartz. Some of the MEMS or biochips which are used for external diagnostic purpose or reaction purpose, those you can make use of quartz or glass or silicon, etc. But the problem comes if you want to implant some of the chips into the living body or living animals or human body, whatever it is. Now, biological entities, those are cells, proteins and DNA. Polymers is most favorable material for BioMEMS application. Some of are polydimethyl siloxane, that is PDMS, another is methyl methacrylate, which is PMMA, Teflon. These are some of the common polymers which are used in biological MEMS. And the commercial players on biological MEMS are Nanogen, who are producing chip based assays on chip DNA hybridization and optical detection, Cepheid DNA detection on a microscale for diagnostic, military and food safety applications, Affymetrix chip based assays on chip DNA hybridization and optical detection, HP and Calipar chip based capillary electrophoresis, Motorola opened a biochip division separately, Hitachi, IBM, TI, HP, they are in the line and they are concentrating on DNA and protein chip. Now, I will discuss the few applications of BioMEMS. First, I will attack on biomechanics, microsensors for biomechanics. So, for biomechanics, one sensor is very much used, that is a strain gauge. Strain gauge is basically made using piezoresistive mechanism of the silicon sensor. Silicon is a piezoresistive material, you know, that is basically used to characterize the forces in the body motion. In the body, in muscle, there are a lot of mechanical movement of the muscles, and if you want to monitor the movements of the muscle, so you need the strain gauges. So, strain gauge, if you attached on any muscle, and then because of the forces in the muscle, so the strain will be developed, and that if it is a piezoresistive, so that can give certain output. Another application of the strain gauges are orthopedic research and the study of muscles in between cellular level, particularly muscles which are in between cells, those characterization is very important in many cases, and in between cell muscle movement and their characterization is possible with micro miniaturized strain gauges, which can be implanted in the particular location. So, MEMS miniaturization has made it possible to make the micro strain gauge, which can be implanted in different locations, even in the muscles which are in in between cellular cells. Third application is understanding intercellular muscle function, which would allow the development of improved locomotion therapies and prosthetic devices. Locomotion therapy and prosthetic devices, for that you need these strain gauges. Accelerometers, that is also used in biological application. Accelerometer, how it is made, already I discussed in detail. These sensors are useful to determine impact level and patient posture. Impact level, suddenly you are hitting on a certain muscle, what is the impact? There is sudden impact, and that can be sensed by inertial sensors. And what is the attitude of the posture of the patient? If you give an impulse on a particular muscle or particular section of any of your body, so that can be properly diagnosed with the help of the accelerometers or any gyros. Both the sensors are used in case of the biological application. Now, microsensors for pneumatic biosystems, they use pressure sensors. We know that human body is a complex system of pumps, valves, vessels and interconnects. Heart is a example of pumps. Valves are there, lot of valves are there, you know, during passage of your fluids, which may be blood also in the human body. Interconnect and vessels means tubes, the arteries and veins, lot of those are interconnect of the fluids. Even in urology, there are lot of interconnects and vessels are there for culturing urine and for disperse the urine. Not only that, the water is also extracted in some cases. Those cases they require the pressure sensor. Wherever is a pump, where is the valve, what is the pressure generated from the pump or valve, that has to be monitored. For that you need pressure sensor. Health monitoring of a patient require knowledge of blood pressure, bladder pressure and cerebral spinal fluid pressure. These are the three important aspects: blood pressure, bladder pressure and cerebral spinal fluid pressure. Those can be precisely monitored with the help of high precision pressure sensors. Pressure sensors are inserted into the body that must be very small and is to be disposable, because once you use in a certain section to measure the pressure of a bladder, the same sensor cannot be used in other place, which is say for example for measuring the cerebral spinal fluid pressure. So, those kinds of sensors must be disposable kind of thing. In this aspect, in a pneumatic biosystems, Lucas Nova sensors are going far ahead for marketing devices, and in this picture you can see the pressure sensors of different sizes, and some of the sensors are highly miniaturized, so that easily you can implant those sensors even in cerebral spinal fluid channel location. So, the sensors of smallest size of the order of 175 micrometer by 700 micrometer by 1000 micrometer is reported from the Lucas Nova sensors, and those kind of sensors can be inserted into catheter and into arteries. So, in the tip of the catheter you can put those miniaturized pressure sensor, and you can push the whole thing into the arteries, because its dimension is negligible in comparison to the channel inside the arteries. So, those are some of the application of the pressure sensors in pneumatic biosystems. Now, coming microsensors for chemical biosystem. Living organisms are extremely sophisticated and they are extremely high sensing molecules, living organisms. And if you want to make some reaction with the with some chemicals, so you have to do it with lot of precautions and care. Lot of applications in medical diagnostic instruments are there for the chemical biosystems. They are drug screening, implantable sensors for prosthesis and environment monitoring. Challenges lies here the delivery, reaction control and waste disposal. For example, if you want to diagnose something and you want to implant something into the body, so major problem is delivery, because how the chemicals will be delivered into that particular location. Then after delivering these chemicals into a living organism, then how can you control the reaction? If the reaction is not favorable to the patient, then you have to control it, you have to stop it. How can you do it? That is another challenge. And third one is waste disposal. Since it is a chemical reaction with the living organism, some byproduct will be there. If that byproduct is not friendly to the human body, how can you remove those byproducts? That is waste disposal. That is another problem. In this direction, for chemical biosystem, some sensors are used which are resistance type impedance sensors. And what is impedance sensor? Those are basically the impedance, or rather the resistances will change with absorption of certain chemicals into the living organism. Gases or vapors and their relative concentration can change the conductivity of some materials, for example, conductive polymers and metal oxide. There are certain polymers, I can name one polymer also which is polypyrrole. That polymer is a conductive polymer and it is directly used as chemi-resistor. There are two things happening in case of polymer if gas is absorbed. One is the conductive polymer, its conductivity changes if gas is absorbed, and the second is if some gas is absorbed by polymer, it swells. Its swelling means its volume changes. Now you see there are two kinds of the mechanisms you can get from here. Once you can, from the conductive change or resistance change of the polymer, directly you can reflect into your voltage signal, and by absorption of certain chemicals or gases or vapors, if it swells, then you can make a capacitance also, and then you have to have not conductive but insulating polymer as a dielectric, and if you have conducting polymer on both sides as a electrode, then on absorption of gas, if it swells, then automatically the capacitance will change. So in both directions people are working and they are making lot of sensors which are used as a gas sensor. One, because lot of gases are there evolved in your human body, in system also. So you have to sense that gases. But the problem, there are two problems. One is the type of gases, which gas is evolving, and another is the concentration of gas molecules. Two things are important. In some cases, the concentration can be directly measured with the swelling, how much swelling in capacitive type of sensors, how much it swells with concentration. And the second, because if you want the selectivity, that means which gas is evolving, then you need the knowledge of polymers which reacts with which gases. In those cases, single polymer sensor may not be useful. You have to have a array of sensors for different kind of polymer, and you have to know beforehand which polymer can react with which kind of gases. So accordingly, both the things are available, which kind of gas and how much concentration of gas is available with this kind of polymer based gas sensors. Now, many polymers will geometrically swell reversibly when exposed to a certain gases. Swelling to a greater or lesser extent depends on variety of gases. Now, one important point is mentioned here, swell reversibly. It is not that once it swell, expand, it cannot contract. Both should be possible. When the gas concentration is more, it swells. When it is less, it may contract. Swells means it may expand or it can reduce the volume. So both are required. A conducting polymer, as I mentioned, polypyrrole is used for this purpose. Insulating polymers are doped with conducting particles like carbon black to reduce their conductivity. Basically, you can start with an insulating polymer, and by adding or implanting many of conductive particles like carbon black, you can reduce their conductivity also. The resistance of doped polymers will change as a function of chemically specific and concentration dependent swelling. One is chemical specific, other is concentration dependent swelling. You see, both things are important. One should be chemically specific polymer, another is concentration dependent swelling, so that you can know type as well as concentration both. Now, some of the sensors which has been reported by Zudi et al. In 1999 are shown in this diagram. Certain diseases cause the body to generate specific gases that are not normally present gas. Normally those gases are not present, but certain disease can generate those gases, and gas sensors of this kind may help diagnose particular patient's health. A response pattern, what is the response pattern? One is conductivity change, another is swelling. These are the response pattern of different polymer is needed to identify specific gases. So, if you want to identify specific gas, you have to have the response pattern beforehand, otherwise you cannot identify the type of gases. Capacitive detection of swelling may also be used for detection and concentration of gases, and that I just told you. You can SU8 photoresist. You see here you can make a group, and in that group you can make the solvent, which is the basically polymer solvent, and if you want to change its conductivity, you can put some carbon black polymer film on top of the oil which contains the solvent. So, these oils are micro oils are created to have to contain the solvent and to react with the gases. So, these are some of the gas sensors. Now, I will talk little bit on electrochemical sensors. And electrochemical sensor basically oxidation reduction of chemical species on a conducting electron is done there, that is detected, and there the oxidation reduction of the chemical species, what will happen due to that? Charge will be generated and the movement of the charge, and if the charge moves, they can be sensed by two ways. One is known as a potentiometric way, which basically potential dependent on chemistry, and the another is amperometric way, current generated by reaction. There are two kinds of sensing. One kind is known as potentiometric sensing and other kind is known as amperometric sensing. Micromachining processes can be used to accurately and reliably define the area, number and relative position of electrodes that are exposed to the solution. So, here comes your micromachining technology with specific, because here you are going to have electrodes and those electrodes are inserted in a chemical where a reaction with the chemicals, those reactions are oxidation reduction reactions and their charges will move from one electrode to other electrode and the movement of the charge will either reflected to the voltage change which is known as potentiometric sensing and also it can be reflected to current change which is known as amperometric sensing. So, these are the basic principle of the electrochemical sensors and here your requirement is to accurately design and fabricate the electrodes, micro electrodes. And some of the examples in this direction are glucose oxidase enzyme to detect glucose. In a blood, how much glucose is there, that can be easily detected by this technique using oxidation reaction using some enzyme. So, this is known as glucose oxidase enzyme to detect glucose. Here the enzyme which is typically immobilized, that is immobilized on or near electrodes, it reacts with glucose and alters the local pH value, oxygen concentration and hydrogen peroxide concentration. So, these can be used for glucose sensor. Those events can be electrochemically detected, that is glucose oxidase enzyme reaction. Next kind of sensor I would like to discuss is molecular specific sensor. One example is ISFET. What is that? Ion sensitive FET. Field effect transistor, you know, source, drain and gate, these three are the electrodes. So, now the performance of the MOS device depends greatly on the MOS on the gate electrode, this voltage on the gate electrode. Now, in this kind of the ISFET, what is being done, the gate is not normal the silicon gate or polysilicon metal gate. Gate is ionic gate. Basically, an ionic solution, they act as a gate. Ionic solution means it will have lot of ions. If the ions changes, their concentration changes, automatically it will have great influence on the performance of the MOSFET. In earlier, we used to apply certain voltage on the gate electrode. Instead of that, we are using a chamber which contains some ionic solution. So, that ion means some charges will be there on the gate and that is going to influence the source to drain, drain to source currents in a MOSFET. So, that ionic solution acts as a gate of an FET which makes the device sensitive to the overall ion concentration of the solution, not selective to specific ions in that case, because depending on the concentration of the ionic concentration in the solution, you can get the drain currents and other things. It can be used a good pH sensor and already pH sensor is made using this technology, ISFET technology. But we are interested not only the concentration of ion, we are interested also to detect the specific ions. How can you do that? That is also possible, which is known as ion selective FET. For that, what you have to do, you have to coat the gate of the FET with certain compound and that compound will attract selectively or bind some of the molecules or ions from the solution. Then it will be ion selective. That means, if you can make the gate with an electrode which is coated with certain compound, and those compounds selectively bind the ions from the ionic solution. Then if these ions are not bind to the gate electrode, so then that is not going to influence the SFET current. So in that way you can make this SFET ion selective also, but the main challenges here are drift and repeatability in this kind of SFET sensors. So SFET is a very popular device in molecular specific sensor. Now another molecular specific sensor is a resonant sensors. Examples are SAW, surface acoustic wave sensors, to detect liquid density and viscosity of the fluid. The resonant sensor are also used as it is made biocompatible, so that it can be directly used with the microorganism and ionic solutions, biofluids. It can be directly used. And what are the resonance structure? The resonance structure will have certain mass also, some flexures, or those flexures may be membrane, or it may be cantilever, or it may be beam. These are the resonating elements. Those resonating elements, what you can do, you can miniaturize those elements, and those elements can be used to selectively react with certain ions. You can coat those resonating elements with certain compound, and that compound will selectively bind to only one specific ions or molecule. Then what will happen? If those resonator will attract some specific ions or molecules, then the mass of that resonator will increase, and because of the change of mass, its resonance frequency also will change. So that is the principle used in this kind of resonance sensors, where we can fabricate accurately some resonating structures, and those structures may be membrane, may be cantilever, may be beams. Now those beams or cantilevers are coated with certain compound. Those compounds will attract certain specific ions or molecules, and when these are attached or bind on the surface of those micromechanical structures, then automatically its mass will increase, and as a result of which the resonance frequency will change. So that is the mechanism which is used in this kind of the resonance sensors which are used in biomedical application. Ion concentration dependent mass loading can be determined by measuring the corresponding shift in the resonance frequency. That is the basic principle. Here the challenges are temperature sensitivity, ion selectivity, reversibility of the binding, and mass loading process. Reversibility of the binding is a major problem, because once it attached to a particular electrode, how can you detach it? So that is the problem. So those challenges you have to sort it before practically use of those sensors in biological or biomedical application. Now microsensors for electrical biosystems. Primary electrical biosystems of interest are central and peripheral nervous systems. These are of primary interest. Sensors and probes have been used to measure the electrical signals generated by neural tissues. Neural tissues, they generate electrical signal, and those signals are sensed or probes using certain microprobes. And here the examples in this direction are electrocardiogram, which is known as ECG, electroencephalogram, which is known as EFG, electroneurogram, which is known as ENG, electromyogram, that is known as EMG, and electroretinogram, that is known as ERG. So, these are various kinds of sensors which are based on electrical biosystems. They are ECG, EFG, ENG, EMG and ERG. Now, some of the examples are shown here. Bioelectrical signals are typically transduced with either internal or external electrodes. In what way MEMS are coming into the picture? MEMS are coming into the picture in this particular cases in fabrication of microprobes or microelectrodes on a single substrate, which can penetrate the tissue without disturbing the tissue. So, here you can see some of the pictures, the array of microfabricated silicon neural probes. So, these tips are, you can imagine, is of the order of few micron, maybe 5 to 10 micron. This length is 300 micron. Now, if you compare the tip with this length, so easily you can see it is of the order of 5 to 10 micrometer only. So, this small tip, this basically electrode, and its tip is micro needle kind of thing, that can easily be penetrated into the probes, into the neurons without damaging it, and you can collect the electrical signals from the neuron for diagnostic purposes, whether it is a retina or it is a heart or is other body. In all those cases, the electrical signals can be tapped with the help of these neural probes, and after that you can process the signal for diagnostic purposes. Another technique which is used sometimes, they use perforated membrane, and those perforated membranes are implanted into into the areas whose or in which place you want to make certain monitoring of, you want to make some, or you want to monitor the neuron behavior or its growth. In that case, if you implant those membrane, in those membranes the regenerated neural tissue will be formed, and they can grow and they can come across through the membranes, small membranes, and which you can easily monitor for your diagnostic purposes. In that area, lot of applications are there. What are the applications are mentioned here? Micromachine neural probes and stimulators are used to control prosthetic limbs with processed signals recorded from the brain or spinal column. Microprobes can be tailored to the physical and temporal dimensions of individual cells. That means, depending on your application, you can tailor the size of the probes also. Neuroscientists can now realistically envision sensing devices that allow real-time measurement at cellular level. At a cell level, these probes can get the signal. So, you can get the real-time measurements at the cell level easily using these microprobes, which are made using micromachining technology. Information from such sensors could be monitored, it could be analyzed and used as a basis of experimental or biomedical intervention. So, these are the applications of microprobes. Now, I will discuss on microactuators. Some of the sensors which are used in biomedical application I told you. Now, let us see how the microactuators are formed and how they are used in biomedical scenario. Biomedical actuators are used to control the biological objects or their environment on microscopic scale. First example is micro manipulators. What are the job of the micro manipulators? Micro manipulators are driven by a micro actuation mechanism capable of operating in a conductive solution. So, that means micro actuation mechanism. How this micro actuation mechanism can be achieved? For them, good candidates are magnetic, pneumatic, thermal and shape memory alloy. These are good candidates for micro actuation in micro manipulators. And now some pictures are shown in this view graph, which are micro actuators. First one is basically is a magnetic micro actuator, which is used for manipulating a single cell protozoa in saline water. Is a magnetic micro actuator, is again taken from Judy 1995. He is from University of California, and they are pioneering in this particular magnetic micro actuator research. In other is micro gripper, which is used for surgical application, microsurgery and any kind of surgical application. Grippers are important tools, and they are used or they are made using shape memory alloy, shape memory forces. This particular micro actuator is capable of grasping tissues during endoscopic surgical procedures. So, endoscopic surgical operation, this kind of micro actuators, which is based on shape memory alloy principle, is nowadays used in operation theatres also. Some other surgical micro instruments are also discussed. The capability of most micro actuators to surgically interact with biological tissues is hindered by their inability to withstand forces on the scale of 1 millinewton, because those micro actuators and manipulators, they are not capable of higher forces. If you have some gripper or some tweezers, so they should be they should be capable of holding something, grip something with more force. But most of the cases, at the beginning we found that the problem is the generation or withstanding more forces, that is not more than 1 millinewton. Recently, high force small displacement stepper motors are used, or resonant micro actuators are used for micro actuation in surgical devices. One is small size stepper motors or resonant micro actuators. So, there you can have larger force if you work in the resonant mode. That is one of the solution people are looking for. MEMS technology offers a variety of capabilities to surgical micro instruments, for example, micro heaters, micro sensors, fluid delivery, fluid extraction, feedback and control system. In those cases, basically the MEMS devices are coming into the picture in surgical environment. Now, here again some of the pictures which shows some of the micro instruments. In the left side you can see one scalpel. That scalpel is basically driven with the piezoelectric forces, and the piezoelectric stepper motor allows the scalpel to precisely control. This is one of the scalpel used in microsurgery, and other picture shows some of the cutting tools which are used in microsurgery, and those are based on ultrasonic vibration. The micro machine ultrasonic cutting tools they are fabricated from bulk micromachining. Piezoelectric material is attached to the cutter to resonate the tip of the tool, because for resonance here you have to have some piezoelectric material which will be resonating by application of certain electric field. So, those piezoelectric materials are coated at the tip of this cutting tool and they are forced to vibrate in with the help of ultrasonic source, or ultrasonic energy is given to those tools so that it can vibrate with ultrasonic frequencies. And nowadays the cutting tools are available using this technique, made using this technique. They can even cut tough tissues like cataract in your the optical or in ophthalmology they are highly used for removing any cataracts. They are basically hard lens, they are lens basically, it is made of hard tissues. Those tissues can be cut using the ultrasonic cutting tools. So, these are some of the applications towards the surgical and micro actuators I just mentioned here. Others are again some kind of valves are shown here, some kinds of needles are also shown here. You can see the diagram in the left side, these are the conventional needle which is nearly 30 gauge needle. You can see here, so in center there is a hole and through that hole some fluid can come, and this is the tip of the needle. And in the right side itself the silicon needles are there, you can see the tip in silicon needle, and if you compare this tip along with the conventional tip, you can imagine how nice needles can be made using silicon micromachining. These are the micro needles, and the right side you can see the micro pump. This is driven by electrostatic energy, electrostatic micro pump with two one-way check valves. This is the inlet, this is the outlet. What is the principle of operation here? And that is electrically driven. That means you have to have the two electrodes here, and those electrodes either depending on the supply they can attract, this is basically a diaphragm. By electrostatic energy this diaphragm, middle diaphragm can be attracted towards the upper diaphragm, as a result of which this chamber, the pump chamber, will be at a low pressure. So, automatically due to the outside pressure the valve will open and the fluid will flow into the chamber. Now, if you release the diaphragm by again the electrostatic actuation, by then this diaphragm will go down and it will put pressure on the fluid into the chamber. So, as a result of which this valve will close and this valve will open. So, if you pressure from inside, this valve will open and the fluid can flow here. So, that means in one valve you are sucking the fluid and other valve you are ejecting the fluid. So, just by using some the electrostatic actuation, it is made using three pieces, top piece, middle piece and bottom piece. There are four pieces, 1, 2, 3, 4, and they are combined together by the wafer bonding technology. And this kind of the valve, micro valve or micro pump is used for sucking fluids during the surgical operation and some other cases. Here some of the micro valves are shown, which is designed by Redwood Incorporated. This is commercially available, some of the micro valves which is used in biological application, biomedical application. So, here I can just conclude that the biomedical application is really fascinating in case of MEMS and microsystems. I have not covered here the DNA synthesis and gene sequencing that required a thorough knowledge of the microorganism as well as the molecular chemistry, biochemistry, and without the adequate knowledge of the biochemistry or cell biology, you cannot go for that. Anyway, some of the idea I gave you in different areas where the biosensors are made using micromachining technology. It may be inertial sensor, it may be chemical sensor, it may be gas sensor, it may be some neuron sensor for microprobes, microneedles, and also I mentioned some of the microactuators which is used in case of microsurgery. So, with this let me stop today.
(1951). A young nation aspiring to find ways to fulfill a dream lays the foundation of an institution that will give aspiring technocrats the license to fly high. The first Indian Institute of Technology is born at Kharagpur. Founded on the basis of the recommendations of the N.R. Sarkar Committee that was set up in 1945 to consider the development of higher technical institutions in India, the institute was first established in 5 Esplanade East, Kolkata, before it moved to Kharagpur in 1951. With Sir Gyan Chandra Ghosh as the first director and Dr. B.C. Roy as one of its founding guardians, the institute established itself as the symbol of a young, dynamic and resurgent nation. As top students rubbed shoulders with the most celebrated of professors and scholars, visions took shape. And IIT Kharagpur continued to play the pioneering role that was envisaged for it, enabling India to become a knowledge powerhouse that it is today.